Welcome to the Induced Innate Immunity module. In this talk, we're going to learn about toll-like receptors. We will begin with learning about the domain structure of toll-like receptors, followed by their cellular localization. We will then learn about signaling pathways and the outcome of these signaling pathways that are initiated by the toll-like receptors. Now, some proteins and cells that play critical roles in innate immune defenses are always present in the host body. These include proteins like lysozyme, defensins, or complement proteins. Thus, they are constitutively present. We also have cells like monocytes and neutrophils that play crucial roles in innate immune responses, and they are also always present in the host body. However, when there is infection that occurs, then some proteins have to be made in response to that infection. And thus the expression of these proteins have to be induced. Usually these are proteins that are involved in inflammation. And their expression has to be induced because if these proteins are present all the time, or if there is constitutive expression of these proteins, then it is actually detrimental to the host. Inflammation normally occurs when the host is able to recognize pathogens that are present inside. The role of inflammation is to direct molecules as well as cells to the site of infection, and these molecules and cells will then play an important role in clearing the pathogen. Another role of inflammation is to activate the immune cells so that they do a better job at clearing the pathogen. Inflammation also induces localized blood clotting, and this helps in curbing the spread of the pathogen to other parts of the body. And finally, inflammation also helps in repairing injured host tissues. Now, inflammation is triggered by the production of different molecules like cytokines, chemokines, and other types of mediators like histamine. The host is able to recognize microorganisms that cause infections by recognizing molecular patterns that are present on these pathogens. These patterns are called as pathogen-associated molecular patterns, or PAMPs. The PAMPs are recognized by molecules made by host cells, which are pattern recognition receptors, or PRRs. Now, the pattern recognition receptors are not only able to recognize PAMPs, but they're also able to recognize damage-associated molecular patterns, or DAMPs. And these patterns occur when there is damage to tissues. The pattern recognition receptors can be soluble molecules like ficolin or mannose-binding lectin, but they can also be membrane-bound or intracellular molecules. The pattern recognition receptors that are present in host cells come under different families. Some come under the toll-like receptor or TLR family of proteins, while others come under the NOD-like receptor or NLR family of proteins. We do have pattern recognition receptors that belong to the RIG1 family of proteins, as well as the STING family of proteins. Toll-like receptors were initially identified in Drosophila, and at that time the protein was called as toll. Now, flies that were deficient in toll were shown to be very susceptible to fungal infections, and that is what we can see in the figure where that white arrow is. The white arrow is pointing at all the fungi that have covered the body of that fly. Now, toll-like receptors were then identified in higher organisms, and now we know that there is an entire family of proteins that come under the umbrella of toll-like receptors. Toll-like receptors represent an ancient pathogen recognition system. In the case of humans, there are 10 TLR genes, and each TLR protein that arises from these genes recognizes a distinct PAMP. So here we can see the different toll-like receptors that are found in humans, including other mammals. And they all are able to identify distinct pathogen-associated molecular patterns from bacteria, viruses, and even fungi. The distribution of toll-like receptors is wide, so we can find them in many different cell types. When we look at the structure of toll-like receptors, they have a leucine-rich repeat domain and followed by a transmembrane domain that helps them anchor to the membranes of cells. On the cytoplasmic end, they have the toll IL-1 receptor domain, which is also called as the TLR domain. Now, pathogen-associated molecular patterns, which function as the ligands for toll-like receptors, are recognized by the leucine-rich repeats. The TLR domain, on the other hand, activates signaling pathways when the toll-like receptor binds to its pathogen-associated molecular pattern. When we look at the localization of the toll-like receptors, they can be present either on the cell surface, and when they are present on the cell surface anchored to the plasma membrane, they are able to detect pathogen-associated molecular patterns that are present in the extracellular environment. You can have toll-like receptors that are present on the endosomal membranes, which are inside the cell. And these toll-like receptors are able to identify intracellular pathogens that are in the endosomes, and these include viruses and internalized bacteria. Now, what should be remembered is that most toll-like receptors are membrane proteins. However, there are some of them that are soluble and are found in bodily fluids. However, they're almost never found in the cytoplasm of cells. When the toll-like receptors are able to bind to their pathogen-associated molecular patterns, it activates a signaling pathway that can result in many different outcomes that include the production of inflammatory cytokines or chemotactic factors. Additionally, TLR signaling can also induce the production of antimicrobial peptides as well as antiviral cytokines that help protect cells from viruses. Now, the binding of the ligand results in the dimerization of the toll-like receptors, and this brings the cytoplasmic TLR domains of the toll-like receptors close to one another. This results in the recruitment of adapter proteins, and there are many adapter proteins that are essential in TLR signaling. MYD88 or MYD88 is the most common adapter protein in TLR signaling. When we look at the table, we can see that with the exception of TLR3, all the other TLRs recruit MYD88 when they get activated. The adapter proteins play an important role in the signal transduction process by recruiting other proteins that help pass the signal into the cell. Now, TLR signaling does aid in the expression of genes, but however, this can happen by the activation of transcription factors like nuclear factor B, activator protein 1, and the interferon regulatory factor family. So let us see how the signal transduction pathway works. The TLR signaling pathway is initiated when a pathogen-associated molecular pattern, which is the ligand, is able to bind to the leucine-rich repeats of the toll-like receptor. This binding induces dimerization of the receptors and brings the TLR domains in the cytoplasm close to one another. These TLR domains can then form binding sites to adapter proteins like MYD88. Once MYD88 is able to bind to the TLR domain of the toll-like receptors, it forms recruitment sites for other proteins like the kinases, which are IL-1 receptor-associated kinases or IRAKs. Now, this interaction is usually mediated by death domains that are present in MYD88 and the IRAKs. The IRAKs that are now associated with MYD88 are able to recruit another protein called as TRAF6. TRAF6 gets phosphorylated and becomes active. TRAF6 is a E3 ubiquitin ligase, and hence it can form ubiquitin chains. The activated TRAF6 can now create polyubiquitin chains that are shown as those yellow triangles. And these polyubiquitin chains can form binding sites for other proteins. Now, in the TLR signaling cascade, proteins like TAB1 and TAB2 are able to bind to these polyubiquitin chains and bring another kinase called TAC1 close to the IRACs. This causes TAC1 to be phosphorylated by the IRACs and activates TAC1. Now, what TAC1 does is that it's able to phosphorylate other proteins, and another complex of proteins called as the inhibitor of kappa B kinase get recruited to the polyubiquitin chain so that now they are closer to TAC1. The activated TAC1 is able to phosphorylate the IKK complex because these proteins are now close to each other due to their binding to the polyubiquitin chains. The phosphorylated inhibitor of kappa B kinase becomes active and can phosphorylate the protein inhibitor of kappa B. The phosphorylated inhibitor of kappa B, or I-kappa B, can now be ubiquitinated, and the ubiquitinated I-kappa B is sent to the proteasome for degradation. This leaves NF-kappa B to be free and able to go to the nucleus where it can bind to the promoter regions of different inflammatory cytokines and induce their transcription. TLR signaling can also activate other transcription factors in addition to NF-kappa B, like AP1. For the activation of AP1, we need to go back to TAC1, which we saw is able to activate the inhibitor of kappa B kinase called IKK. Now, TAC1 can also phosphorylate MAP kinases, and these MAP kinases can then activate proteins called FOS and JUN. FOS and JUN can then activate AP1, and AP1 is a transcription factor that can bind to the promoter of genes and induce the expression of inflammatory cytokines. Some members of the toll-like receptor family are able to bind to RNA. One such example is toll-like receptor 3 or TLR3 that is present on the endocytic membranes of a cell. Now, TLR3 is able to recognize and bind to double-stranded RNA, and this double-stranded RNA is normally found in viruses. The binding of TLR3 to double-stranded RNA results in its dimerization, and thus the TIR domains of these two TLR3 molecules are close together in the cytoplasm. The TIR domains are able to recruit the adaptor protein called as TRIF, which in turn is able to recruit the E3 ubiquitin ligase called TRAF3. TRAF3 gets activated through this interaction and is able to form a polyubiquitin chain that functions as a scaffold where other proteins can bind. The binding of a complex of proteins to these polyubiquitin chains results in the activation of the kinase TBK1. An activated TBK1 is able to phosphorylate a transcription factor called as interferon regulatory factor 3 or IRF3. IRF3 gets dimerized once it is phosphorylated and is able to enter the nucleus. Once it's in the nucleus, the phosphorylated IRF3 is able to bind to the promoter regions of DNA that code for type 1 interferons, which include interferon alpha and interferon beta. These type 1 interferons help in protecting host cells from viruses and hence play important roles in an antiviral response. Some TLR family members are able to recognize and bind to single-stranded RNA. One such example is TLR7 that is present in the endocytic membranes of host cell and is able to bind to single-stranded RNA. The binding of single-stranded RNA to TLR7 is able to activate TLR7 by promoting its dimerization. Now, the TLR domains that are close to one another in the cytoplasm are able to recruit the adaptor protein MyD88, which in turn is able to recruit the kinases called as IRACs. Now, the IRACs can interact with a transcription factor that is present in the cytoplasm called interferon regulatory factor 7 or IRF7. And this IRF7 gets phosphorylated by the IRACs. The phosphorylated IRF7 is able to form a dimer, and this facilitates its entry into the nucleus, where it is able to bind to the promoter regions of type 1 interferon genes, which include interferon alpha and interferon beta. These type 1 interferons that are produced help in generating an antiviral response that protects host cells from viral infections. Thus, we looked at how TLR signaling helps in promoting an inflammatory or antiviral response. To summarize, we have toll-like receptors that are mostly membrane proteins and are present either on the plasma membrane of host cells or in endocytic membranes inside host cells. When these toll-like receptors bind to their appropriate pathogen-associated molecular patterns, they get activated and undergo dimerization. These dimerized TLR molecules are able to recruit adapter proteins to their TLR domains in the cytoplasm of the host cell. The adapter proteins are able to recruit other proteins like IRACs, which in turn recruit TRAF proteins that are able to form polyubiquitin chains. These polyubiquitin chains are able to activate kinases that result in the activation of transcription factors. Such an example is the activation of the IKK complex, which is the I-kappa B kinase complex. When they get activated, they are able to phosphorylate I-kappa B. And once I-kappa B is phosphorylated, it can get ubiquitinated and degraded by the proteasome. This allows NF-kappa B to become free and enter the nucleus, where it is able to bind to the promoters of genes that code for inflammatory cytokines. Additionally, we can have MAP kinases that get activated due to TLR signaling, which result in the subsequent activation of the transcription factor AP1. AP1 is also able to induce the transcription of inflammatory cytokines. Another way that toll-like receptor signaling is able to protect host cells is through activating the interferon regulatory factor member of proteins, and these proteins are transcription factors that can induce the transcription of the interferon genes. And thus we can have IRF3 get activated or IRF7 get activated, and these activated IRF proteins undergo dimerization, where now they're able to function as transcription factors as they enter the nucleus and induce the transcription of type 1 interferons. To summarize, we learned about toll-like receptors in this talk, which includes their domain structure, cellular localization, signaling pathways that are initiated by the toll-like receptors, as well as the outcome of TLR-mediated signaling.